Yep,
Okay,
looks great.
Perfect, thank you. So thank you for the lovely introduction. I'm thrilled to be able to speak to this particular group today. And of course, every time I put together this type of an overview talk where we're thinking about genomic analysis, the list of things that I want to speak about or that I want to make sure the audience knows about is far longer than the time that we have to speak about it. So I want to, I guess, just reinforce that this is very much meant to be an overview and a starting point. I think that, there will be certain topics that will be covered a little bit more deeply later on today and on day 2 and on day 3. And then I also anticipate that as many of you are working through the demo cases, you'll also have the opportunity to start to use some of these resources. And as you dig in, you may have very, very specific questions. So with that in mind, I wanted to cover a little bit of a broad array of things. I'll start just with a few comments on analysis platforms and data storage and how to think about choices related to those issues, and then move on to what I would call analysis support tools, tools that we can use to identify existing or known gene phenotype relationships, or genotype or allele phenotype relationships. So, specific to a particular variant identified. Think a little bit about minor allele frequencies across populations, matchmaking, and the ClinGen allele registry. And so I'll start first just with a little bit about commercial versus open source platforms, because sometimes this is a bit of a question that, that comes up. So I made just a little bit of a sort of a fairly simple comparison table. Typically with open source platforms, you wouldn't need a license to be able to use them. And so you wouldn't need to buy a license. So they tend to be free in terms of financial cost. Whereas with commercial platforms, there's often a cost involved, and you typically do need to purchase some type of license. Open source platforms for genomic data analysis are often a little bit easier to customize, although it may be on you and your team to do those customizations unless someone else in the general community has already built the customizations that you need. With commercial tools, customization either may not be possible or may depend heavily on the vendor and the particular tool that exists, and whether they're interested and able to sort of further develop that specific to your needs. In terms of sort of support and training with open source software, a lot of that really comes from the community. You may be able to link back directly to the person or the team that created the particular platform that you're seeking to use or that you are using, or you may be able to connect to other users through blogs, chat groups, kind of online venues such as that to ask questions and to get support and information. Whereas with commercial platforms, you tend to need to rely on vendor support. The other thing to think about is viability, and this can really vary incredibly widely. If you're wanting to use a platform that you expect to be still in use and perhaps further developed over the next 10 years, that development may be a little bit more stable if you're working with a commercial platform, assuming that the vendor continues to do well. With open source, it's very dependent on the open source provider. Have they kind of provided a one-time platform and then walked away to other things, or is this a platform that they're using in their own work and continuing to develop and to add to, or could you even become, partner with them to continue to develop that particular tool or platform? So some considerations when deciding between these, there's really no one size fits all. And so what I wanted the take home for today to be is that you very much have to consider what your needs are. And so I've provided here 2 scenarios that I think are very different and that might lead to very different decisions about ideal platforms for genomic analysis. So scenario A, Dr. M is a medical geneticist. She wants to analyze negative exome data from patients she's seen in clinic. She's expecting to analyze about 1 or 2 cases a week. She really doesn't have any unique analysis needs. She's a full-time clinician. She's performing this reanalysis on her own in her spare time. You could contrast that with scenario B, where you have Professor R. She's a researcher specializing in molecular genomics. She wants to integrate RNA sequencing data into her research pipeline. She's anticipating 1,000 cases a year. She very specifically wants to integrate those data with existing genome data, so she has a very kind of specific need. And she leads a team of 10 individuals, including some computational experts. So she may have a little bit more bandwidth, either for platform development or implementing and customizing an open source platform. So these are 2 individuals who may end up coming to very different decisions about what's the right platform and overall approach for their needs. Some considerations as you're working through these decisions. What exactly do you need in terms of analysis? We'll talk a little bit more in later sessions about some of these steps. Are you needing to take a raw BAM file or a raw FASTQ file through alignment, variant calling, variant annotation, and then your variant analysis and prioritization? Or do you have an annotated, a VCF file, a variant annotation file, and you really just need to look at the variants and prioritize, think about them deeply? Are there specific customized needs or unique needs for you and what you're trying to do that might be different from what's available in common existing platforms? Along those lines, in terms of what are your needs, what also are your input file types? So you might have very different needs if you're starting with a FASTQ and need to get to a variant call file than if you already have a variant call file that's ready to go. You want to think about what effort is needed to develop expertise in the platform. Is this a platform that's pretty much ready to go, very easy to use, an intuitive user interface? Or is this going to be a platform that is going to take some time just for you and your team to get up and running? And then, of course, always related to that, what is the cost? Hand in hand with thinking about commercial versus open source platforms, many of us think about, well, where are we going to put the data? And I think the common decision point that many of us make at some point or at many points in our careers with different projects is, do we need local data storage? Do we need to use the cloud? And so I wanted to address that a little bit as well. I think this has changed substantially over the last decade. So if you see here with the graph, the cost of cloud storage has decreased substantially, at least with some of the cloud platforms that are commonly used in the US. So we have AWS, the Google Cloud Storage as examples, and you can see that right around 2015, many of the costs substantially dropped. And I think that this has sort of changed a lot of the thinking about data storage for many of us who work with larger data sets. You also want to think about your data size, and I think it's really important to think about not just what data you have now, what are you going to have 5 or 10 years from now? Is this something that needs to be scalable? Larger data sets tend to be more difficult to move around, to download to local servers. If you're trying to use an existing data set, but you want to run analyses on your own server, it can take a while just to get the data there, versus is there a way that you could move your analysis tool into the cloud and analyze the data where they are? Other things that we often think about when we're thinking about data storage, how frequently do we need to access the data? There may be some data types that we say, you know, we can archive this, we might need to get back to it once a year. An example of that for some research and clinical programs might be a BAM file. Once you've aligned your reads, you've called your variants, you have your variant call file, that might be your daily access point, and you might be able to archive the BAM file for sort of a rainy day or a new analysis tool that requires it for input. And so thinking a lot, not just about the size of the data, but how frequently are you needing to access it, and are archival solutions, which tend to be a little bit cheaper, a viable option for you. You absolutely have to think about security and privacy. Certainly research and clinical patient privacy, institutional and human subjects protocol requirements, and these types of issues. And then the larger your team, you have to start thinking about collaboration and access and kind of balancing that. And certainly the ease of sharing data and whether or not there's a possibility for a collaborator to sort of just enter your cloud and analyze data directly there versus the need to sort of transfer it over to wherever they are. So some of these things are really important to consider when you're starting to think about what your basic mode of data storage is going to be. So I want to shift gears a little bit and now start to talk about some of the sort of analysis support tools that I want to make sure everybody knows about as you start these next 3 days. And the first one, certainly many of you may have heard of and used before, online Mendelian Inheritance in Man. This is a tremendously useful online catalog. I've put the website here in the corner for searching genes and Mendelian conditions or rare genetic disorders. And you can identify a few different quick pieces of information by doing that. You can see, and I'll show you that in a moment, but you can see here the search bar, you can enter a gene, you could enter a phenotype, or you could enter the name of a syndrome like Dubowitz syndrome in here. When you do that, if you were to enter a gene name, and I'm showing you here what came up for me when I searched for the gene RET, you can see, first of all, the full gene name, so you can kind of get a sense for what this gene is. You can see the HGNC approved gene symbol. So some genes will have multiple different gene symbols, and you can see which is the approved, but you can also identify what the other names for this gene may have been in the past. And you can also then very quickly see what are the different human disease traits or phenotypes that have been associated with this gene. So you can see with Rett syndrome that it is associated with central hypoventilation syndrome, medullary thyroid carcinoma, multiple endocrine neoplasia. So it's a very quick way to say, I've identified a gene of interest in my analyses. What is known in terms of human disease phenotypes about this gene? Each of these different disease conditions has its own phenotype MIM number, which you can see over here as well. If you were to do the opposite and instead search OMIM for the disease trait or the Mendelian condition, for example, if you searched for central hypoventilation syndrome, you would see this entry here. A lot of the disease traits now have their own symbol. So you can see this is CCHS. So it's not a gene symbol, but it's a Mendelian disorder symbol. And you can see when you look for this particular condition, you get this beautiful table here with a list of the genes that have been associated with this particular condition. And so this can be a very helpful way if you're thinking of a condition to think, what is my genetic differential? What is my gene differential for this particular case? The other thing that's very helpful for kind of pulling up quick information on the fly is the OMIM clinical synopsis, which you can see down here in orange. If you connect or click on this little triangle here, what you'll see is something that comes up a little bit like this. I'm only showing you the top half of this list, but you get a list of the different phenotypic features that have been associated with this particular disease condition. It won't necessarily be an exhaustive list of phenotypic features. So if there's a feature that's only present in 10% of individuals, that may not make it in here. But it can quickly give you a sense if you're, for example, studying a gene that's associated with Phelan-McDermid syndrome, and you say, I don't really know what the phenotypic features of this are, so I can't tell if this matches with the case I'm thinking about. You could start with this OMIM clinical synopsis to start to get a sense for whether that gene is of interest for your case. The next database that I wanted to talk to you a little bit about is Clinvar, and you'll hear more in different ways about Clinvar, I think later on. But this is another very helpful resource for identifying gene phenotype relationships. And in this case, it's actually really genotype phenotype relationships. And in addition, you can find supporting evidence for assertions on variant pathogenicity, which you'll hear a little bit more about on Friday. One of the things that's so helpful about Clinvar is that there are many genes for which distinct variants may lead to different syndromes. And so this can be a helpful place if you're looking at a variant to figure out what's been reported about this particular variant and the disease phenotype it might be associated with. So to give you a sense for what these entries look like, here is one example. This is actually an entry for BRCA1, and you can see the information across the top about the specific variant that I've selected to look at. This particular variant has been called likely pathogenic. And you can see some of the different variant details and variant kind of IDs and ways of looking at the variant here, some of its different names. If you were to scroll down, you would then start to see a table that would show you the different interpretations that had been provided for this variant to Clinvar. And so in this case, I'm showing you 2 rows. They both show interpretations of likely pathogenic, meaning that this variant was felt to meet American College of Medical Genetics and Genomics guidelines for a likely pathogenic status. We'll talk kind of more about that on Friday, exactly what that means and how to get there. You can see that the submitters for these assertions are 2 laboratories, Ambry Genetics, which is a diagnostic laboratory, and the University of Washington Department of Laboratory Medicine, UW. And both of these laboratories have very nicely provided sort of an explanation or evidence details for why they're calling this variant likely pathogenic. Why don't they think it's benign? And you could sort of read this and you can see this sort of extends beyond what's captured here. But you could click on the more, and there's a nice paragraph describing their thinking and what's known about the variant or what was known at the time that they interpreted it. I next want to move on and highlight just a few things from the Nomad database. So this will by no means cover all everything that you can do with this database, but I wanted to highlight a few things to make sure that you know about these, as I think they'll be helpful to you later in the week. So this is the Genome Aggregation Database. And it's a data set of aggregate or summary genomic data. So the source is really large scale sequencing projects. These are exome sequencing and genome sequencing data sets. And this is sort of the main landing page, which you can get to from the website that I've shown here. You could also Google Nomad and get to it that way. And you can search by a gene, a region, or a variant. And when you do, if you search by a gene, here I'm showing you DRD4, you can see a few things at the very top of that come up at the very top of that gene entry in Nomad before we even get to the variant data. First of all, you can see the gene, the primary transcript laid out here with the exon structure. So this is exon 1, exon 2, exon 3, and exon 4. It's a 4-exon gene. You can see then mapped above it in blue the depth of sequencing coverage from the exome data and the depth of sequencing coverage from the genome data in green. As you kind of start to look down towards the bottom of this particular view, you can see also a representation of the variants within this gene that are in Nomad, and different variant types are represented by different colors. The height of the ovals is indicative of minor allele frequency. If you were to continue to scroll down that page to look at what's below this sort of variant representation, you would then come to an actual table of variants, and you can see each variant listed by its variant ID using sort of what I'll call Nomad nomenclature. So the chromosome position, the chromosome, the position on that chromosome, the nucleotide position, the reference nucleotide, and the alternate nucleotide. You can see the consequence of that. You can see the allele frequency and whether or not any individuals in this database were homozygous for that particular variant. And from here, you could click on one of these variants for more information. And when you do that, it takes you to a variant page. At the top, you'll see exactly which variant you're looking at. You get information on allele counts, annotation information, population frequency information, which you can see in the lower left here. So broken down minor allele frequencies broken down by studied populations, also broken down by sex. And then finally, for the particular variant you're looking at, you can see the age distribution. So it's the distribution of what are the ages of the individuals who have these variants. And finally, I won't go into a lot of details about this, but there's a very, very helpful set of constraint metrics that are available in Nomad, and these are by gene. And what they are is they are an assessment of the number of observed compared to expected variants of a particular variant type or category. And these categories are synonymous variants, missense variants, or putative loss of function variants. And the idea behind the big picture behind looking at these data is that you can use the observed over expected ratio, which is the O over E shown here, to get a sense for whether or not this gene is constrained for synonymous variants, missense variants, or loss of function variants. So you can see here for the missense variants, you observe far fewer missense variants in this gene than you would expect based mostly on the size of the gene. This is a very high Z-score. This gene is constrained for missense variation. It's also constrained for loss of function variants. And you can look here at the observed over expected ratio. There's also a calculation of a PLI score, or a probability of loss of intolerance, probability of loss of function intolerance for this gene. When that number is 1, it means the gene is not tolerant to loss of function. When that value is 0, it means it is predicted to be tolerant to loss of function. And there's also shown here in red, the upper boundary of this confidence interval is called the Luff score. And this can often be helpful for smaller genes where your number of expected and observed variants in total is much smaller. And so this provides a much more powered assessment of loss of function intolerance. I next want to move on and talk a little bit about resources that can be helpful if you've identified a particular gene of interest in your family, and now you're looking to vet that. You're looking to identify additional families. Or if you're studying a family with a very specific and unique phenotype and you're looking to identify additional families to add to your cohort, or to share information with others who may be studying those families. And so the Matchmaker Exchange is a tremendously helpful resource in this regard, a worldwide resource. The goal of the Matchmaker Exchange is really to allow users to identify the genetic cause of rare disorders through genotype and phenotype matching with clinicians and researchers worldwide. It uses a federated platform that connects rare disease databases, or what we'll often sometimes call nodes. So you can see these nodes are based really in institutions around the world. And I should go back and say, the way that you can interact with the Matchmaker Exchange, we'll kind of talk later about how to make a Matchmaker Exchange entry. But conceptually, you might, for example, go to GeneMatcher, which is one of the nodes here, or Seeker, which we'll talk about a little bit later in this session, to enter a particular gene that you'd like to identify additional cases that have rare variants in that gene. So to kind of match with others, you can actually, when you enter that gene into one of those nodes, indicate that you would be happy for that query to be shared across the Matchmaker Exchange. And then you would match with anybody interfacing with any of these nodes through the exchange who might have a patient or an interest in rare variants in that same gene. And then you could kind of share information and say, you know, I have a rare loss of function variant in this gene, the phenotype is intellectual disability, do we have a match? So that's how people most commonly use the Matchmaker Exchange in the nodes. I wanted to kind of show you these numbers. These are the different Matchmaker nodes, and these are the exchange statistics, so the number of cases and patients that have been entered into the Matchmaker Exchange, and the number of unique genes that have been entered or submitted to each of the nodes. You can see that there's a tremendous wealth of genes that have been entered, a high likelihood of obtaining a match that you can at least pursue further and decide whether or not you have shared phenotypes and might have two cases that are very similar. And so this can be just an absolutely tremendously powerful resource. I wanted to also mention in this vein variant matcher. So conceptually, variant matcher is very similar to the gene matcher node of the Matchmaker Exchange. The focus here, though, is on identifying research subjects, not only with a shared gene of interest, but a shared variant of interest. This particular tool is developed by the Baylor Hopkins Center for Mendelian Genomics, and it allows you to search by genomic coordinate for your variant of interest. And I've provided the website here. Variant Matcher actually directly communicates with another resource called PhenoDB, and this was one of the database and analysis tools that was developed by the Centers for Mendelian Genomics, so I wanted to just mention this briefly. This is a freely available website, and I've provided the URL. Clinicians and researchers can enter phenotype information in a standardized format, clinical diagnosis information for families, pedigree structure, and they can also then upload variant call files, and there's an analysis module within PhenoDB as well. So here's sort of to give you an idea of the structured phenotype data, which is ultimately coded in human phenotype ontology terms. And then the variant call file analysis module, which gives you a sense of how you might be able to kind of analyze, for example, exome data. And so this is a tool. You'll hear more about another very similar tool, similar in many ways tool, Seeker, and you'll have a chance to kind of practice your analyses on Seeker. GenotoMP is a tool or a database through which you can identify phenotypic profiles that are linked to rare genotypes. So again, trying to match a gene or variant of interest with phenotype. Your search bar is here, and you can enter a gene or chromosomal position or a human phenotype ontology term. And I think off to the side on the main landing page, they have a link so that you could, if you have a phenotype but you don't have handy the human phenotype ontology term, you can actually use that to search and identify the formal HPO term and then enter that term here. This includes a database of over 19,000 individuals. Decipher as the database of genomic variation and phenotype in humans using Ensembl resources. This is another database of genotypic and phenotypic data, 42,000 individuals. This includes with it the ability to be put into contact with the referring clinician, and also a link to their genome browser through which you can identify and visualize variants. And then finally, I wanted to just mention the ClinGen Allele Registry. So this was really designed with the purpose of providing both unique and also stable allele identifiers for different alleles. And so that the ClinGen Allele Registry, or CAR identifier, would be linked to other variant or allele identifiers such as the genomic or transcript alleles, DB-SNP ID, Nomad ID, Clinvar IDs. It solves the problem that we sometimes have when, for example, a transcript or transcript ID changes over time, so that a variant is no longer meaningful when it's associated to a transcript ID that's no longer used. So again, it's meant to be a stable identifier. Here's an example of a CAR ID, CA118639, and you can see that this matches to a Nomad allele, DB-SNP ID, and amino acid change. These were just a few examples. This is actually the CFTR delta F508 allele that's been well studied. So you can use this resource to identify alternative allele names. You could take two allele names and confirm whether or not they actually refer to the same allele or are referring to different alleles. So it can be a very, very helpful resource to use. So with that, I'll go ahead and wrap up my take-home points. No one size fits all for data storage and analysis platforms, but hopefully I've provided you with a little bit of a resource for how to start to think about making those decisions. OMIM and Clinvar are very helpful tools for informing gene or genotype-phenotype relationships. You can identify similar cases, whether it's cases that share variants in the same gene or cases that share phenotypes through the Matchmaker Exchange and its nodes. You can identify cases with the same variant through Variant Matcher, and the ClinGen Allele Registry is a very helpful tool for identifying unique variant and alleles and mapping them to other variant and allele identifiers. And with that, I'll wrap up and take any questions.